Chromosomes clash day and night. Genetics laid bare under light. Bunsen burners flicker bright. All in the name of Mr. Right. Doing science right tonight. Answers never out of sight. Biology battles fierce and bright. Learning hard and feeling right.
Hi, since this is going to be the second PowerPoint on DNA and genetics, there are some external videos in this PowerPoint. I will link them at the bottom on Doing Science Right, because I can't play them in the actual video themselves. First things first, like last week, this is the list of need to know. So at the end of this PowerPoint, these are the 9 things that I expect you'll be able to do, all of these which can be tested upon in your test at the end of the term, or sorry, halfway through the term, and also in the exam at the end of the year. I'll let you read those ones. There is no need to write those ones down, but we will now go into what is a gene. So first off, a gene, to start with, highlighted in yellow there, a gene is a heritable factor that consists of a length of DNA and influences a specific characteristic. So this is just a certain section of our DNA, could be hundreds, thousands of nucleotides long, that is going to code for a specific protein, and then that protein is going to be made and work as an enzyme or to catalyze a reaction. In general itself, genetics, which is the field that we're looking at here, it's a branch of looking at storage of information in living organisms and how we pass that information down through inheritance to our offspring. Generally themselves, when we're talking about passing down, we're going to be talking about chromosomes. Chromosomes for humans and for anything that's a eukaryotic cell or has organisms that have eukaryotic cells. Their chromosomes will be thin, they're linear structures, and they're made up of DNA wrapped around a protein called histones. In humans, there are 46 chromosomes in each of our cells, but there's thousands of genes in every cell because there's hundreds of thousands of genes on each one of those chromosomes. That does mean that a gene is much smaller than a chromosome, and the chromosomes are going to be made up of many genes. A gene itself, like I mentioned at the beginning of this slide, is a sequence of DNA that gives rise to a specific trait, and those traits might be influenced by multiple genes, but each gene itself is going to influence one trait. In terms of those 46 chromosomes that you have, you've obtained 23 from your mother and 23 from your father. Looking at comparisons of genes, how many genes an organism has doesn't technically tell you how complex it is biologically. So we can see, for example, over here that humans have roughly 22,000 genes, but a grape has 30,000 genes. So you can tell straight away that how many genes something has isn't a degree of how complex the organism is, or even of the intelligence itself. When we're doing this and we're trying to figure out how many genes an organism has, there's lots of different approaches they can use. So those final estimations, like humans having 22,000, there will be a large range. So it might be like between 20 and 25,000 genes in the human genome. 22,000 being very specific, and not every person will have those 22,000s. If we're looking for a good example of this is rice, which has a much larger estimation there. Rice, for example, can have anywhere between 32,000 to 50,000 genes, and if we're looking at the correct scientific numbers for humans, that's 19,000 to 25,000. So these are just estimates, but you can still tell that it's not a measure of how complex or intelligent an organism is. Each person, especially humans, because we have 2 chromosomes and there's 23 pairs, we have 2 copies of each chromosome, which means we have 2 copies of each gene. Now those genes can be slightly different, and they have alternate forms. This is what we call alleles. So alleles are just alternate forms of the same gene. A good example of this is eye color. So you might have 2 chromosomes, you might have a gene for blue eyes and a gene for green eyes, and because green eyes is more dominant, your eyes appear green. There are often more than 2 alleles for a gene, so like eye color again. Eye color obviously we've got brown, hazel, green, blue, etc. There's more than 2 alleles, so the combination that you have can vary quite a bit. Because alleles are just the same gene but a different type of that gene, they will have the exact same location on the chromosome as their other alleles, and that location on the chromosome is what we call the locus. only one allele can take up the locus on a gene at a time, so you couldn't have more than 2 of each types of allele. So you couldn't have blue, green, and brown genes for eye color because you've only got 2 locations for eye color genes, one on each chromosome pair. Alleles will generally differ by only a few bases, sometimes only one base. So, for example, the difference in blue and green color. Eye color is not actually the best because it's controlled by 3 or 4 genes, but just as an example, the difference in blue and green eye color could just be one nucleotide base out of hundreds and thousands of genes. So just one mutation, one different base, and you have a different appearance of the final organism. Those new alleles, like different eye colors, are all formed through what we call gene mutation, which is a random change in the nucleotide base sequence. The most common type of base mutation is a base substitution, where one base is replaced by another, which is a mistake which happens in a process called DNA replication. Because of the years of evolution that humans have gone through, or all organisms have gone through, it's very unlikely that a random change to an allele is going to be beneficial to that organism. Most mutations that we experience, in humans at least, are either neutral, so they have no benefit and no gain, no harm, or they are harmful to the organism. Some mutations, such as cancers, are lethal, which will cause death. Mutations in the body, when death occurs, are eliminated, but if a mutation happens and that mutation is passed on to an offspring, that's what we say causes genetic disease. Moving on to genome, so this word genome, this is all of the genetic information in an organism. For us as humans, that consists of all the DNA that are in our chromosomes, so all the DNA in the nucleus, but also DNA that exists in our mitochondria. In plants, it's all the DNA that's in our chromosomes in the nucleus, plus the DNA in the mitochondria and chloroplasts, because plants have chloroplasts and humans don't. Genomes of prokaryotes is much smaller and is only the DNA in the circular chromosome, plus any information that's in the DNA plasmids. We actually made a big effort a few years ago to do the Human Genome Project, which was an international cooperative venture, showed collaboration between many different countries to sequence the entire human genome, so every gene that existed in the human body. It showed, when we did that Human Genome Project, that humans shared a majority of their sequence with each other, and it was finished in 2003 and had a number of different goals that we met by doing the Human Genome Project. One was mapping, so we knew the number of genes, the location of those genes, the size of those genes for all of the human race. So we could then look at and try and figure out where those genetic diseases were, which allowed us to increase screening. By knowing where those genes are, we could increase the screening processes in medicine and try and be able to detect carriers of genetic disease much earlier than previously thought. By knowing the genes that cause some genetic diseases, we can start to create medicines to help them and create proteins, etc., to try and alleviate those genetic diseases. But it also allows us to look at our ancestry and look at where the evolution and the origins and the migratory patterns of the human species. This is the little video. I will link this on Doing Science Right, so a video on what the Human Genome Project is. But more specifically, I'd like to talk about one genetic mutation that is detrimental to humans, and that's through sickle cell anemia. This is a genetic disease that's caused by a one base substitution change. When our DNA is turned into messenger RNA, they transcribe the information wrong, which changes the shape of the protein. This is the most common genetic disease in the world, sickle cell anemia. They change one gene, or one nucleotide, from an A to a T, so the codon GAG is now changed to TTG, which changes the type of protein that is made during protein synthesis, which changes the folding of the protein, which changes the shape of the red blood cells. As you can see here, this is what a normal red blood cell looks like, and this is what a red blood cell of someone with sickle cell anemia looks like. When you have the blood cells of that shape, the cells can become trapped in our capillaries and blocks them and reduces the oxygen flow, which can cause a lot of issues. That's it for this PowerPoint. I will stop it here and we'll discuss more of this in class.